Hi guys, let's do section 4, inside the mitochondria. The preparatory reaction, connects the glycolysis to the citric acid cycle. You'll remember that the product of glycolysis is two pyruvate molecules. Those two pyruvate molecules enter the mitochondrial matrix. That's the inner folded part of the mitochondria. The pyruvate is then converted into a 2-carbon chain called an acetyl group, and then it is almost immediately attached to an enzyme called coenzyme A, and it forms acetyl-CoA. Electrons are picked up by NAD+, producing NADH. Okay, carbon dioxide is released and transported out of the mitochondria into the cytoplasm through the process of making this acetyl-CoA. It is important to get rid of that carbon dioxide quickly, because if you get too much of it in the cytoplasm, it changes the pH, and you'll remember that changing pH is one way to denature proteins. So if we have too much carbon dioxide, we change our pH, which means we have denatured our proteins, and then the cell can't live, it can't do its job properly. So that carbon dioxide is released into the cytoplasm, and then the cell gets rid of it. You'll also remember that 2 pyruvate molecules are made in glycolysis. So for every single glucose molecule, we get 2 pyruvates, which means we get 2 acetyl-coenzyme molecules. So inside the mitochondria, we take a pyruvate molecule, okay, and coenzyme A, okay, and we make acetyl-CoA, which looks like this, and we release 2 carbon dioxide molecules. So here is a diagram showing the structure of the mitochondria. Here is an artist rendering, and here is an actual image taken using an electron microscope. It is a double membrane organelle. That means that it has 2 phospholipid bilayers. It has an inner membrane and an outer membrane. Okay, this space between the two is called the intermembrane space. That makes sense. The inner area of the inner membrane is called the matrix, so that open area is the matrix, and the folded parts are called the cristae. All right, so it's important that you know what those structures are. So let's talk about the citric acid cycle. It's also known as the Krebs cycle. It occurs inside the matrix of the mitochondria, and it begins with the addition of that acetyl-CoA, that 2-carbon acetyl group. It's going to form it into a 4-carbon molecule called oxaloacetate, forming a 6-carbon molecule, also known as citric acid. NADH and FADH2 capture energy-rich electrons, so those electrons that have a lot of energy, and ATP formed by substrate-level phosphorylation. Okay, so we get a little bit of ATP. Okay, this citric acid cycle happens twice because we broke that glucose molecule into 2 pyruvates. So we have 2 molecules that go through this process. And it produces 4 molecules of carbon dioxide, 2 molecules of ATP, 6 molecules of NADH, and 2 molecules of FADH2 per each glucose molecule. So you might be thinking, Ferris, this isn't very efficient. We only got 2 ATPs. That's not a whole lot of energy. Well, you're right, it's not, but we got 6 NADHs and 2 FADH2s. Okay, those are going to be used later on in this process of cellular respiration to make a lot more ATP through the electron transport chain. So here we have another diagram. We have glycolysis happening outside the mitochondria in the cytoplasm. Okay, we got a little bit of NADH that's going to go in to the mitochondria. We then have our prep reaction, where we made a little bit of NADH. We then use the products of the prep reaction in the citric acid cycle, where we get even more NADH and FADH2s, which are then going to be sent to the electron transport chain, where the bulk of the ATP is produced. So the citric acid cycle begins when a 2-carbon acetyl group is carried by coenzyme A, and it creates a C4 molecule, a molecule with 4 carbons in it, and that molecule is known as citrate. Twice over, substrates are oxidized as NAD+ is reduced to NADH, and carbon dioxide is released. ATP is produced as an energized phosphate is transferred from a substrate to ADP. So we put those two together, we get ATP. And then again, a substrate is oxidized, but this time FAD is reduced to FADH2. And then, once again, a substrate is oxidized and NAD+ is reduced to NADH. And we start the cycle all over again, using those enzymes over and over and over again. You'll notice that our inputs in the citric acid cycle are 2 acetyl groups, 6 NAD+, 2 FADs, and 2 ADP plus P. Okay, we use those to produce 4 carbon dioxide molecules, which are then kicked out of the mitochondria and then even further kicked out of the cell. We make 6 NADHs and 2 FADH2s and 2 ATP molecules. It doesn't look like a lot, but these guys are going to generate a lot of ATP in the electron transport chain. The electron transport chain, okay, its location in eukaryotes, so plant and animal cells. It's in the cristae of the mitochondria, and it happens in some prokaryotic cells, some bacteria, in the plasma membrane. The electron transport chain is a series of carrier molecules, and they pass energy-rich electrons from one to the next. It's like a game of hot potato. It's a complex array of proteins and a special one called cytochrome. Proteins with heme groups contain central ion atoms. The electron transport chain receives electrons from NADH and FADH2, and it produces ATP by oxidative phosphorylation. Oxygen is the final electron acceptor, okay, and it combines with hydrogen ions to form water. The fate of the hydrogens. The hydrogens from NADH deliver enough energy to make 3 ATP molecules. Those from FADH2 have only enough energy to make 2 ATP molecules. Those spent hydrogens, the ones that have been used, combine with oxygen that is in the matrix, and they combine to make water. Recycling of coenzymes increases the efficiency. So remember, enzymes are used over and over and over again. Okay, that increases the rate of chemical reactions. Once an NADH delivers its hydrogen, it returns as NAD+ to pick up more hydrogens. It's used over and over and over again. It's like a rechargeable battery. Once it's used, it gets charged right back up. Those hydrogen ions, those hydrogens that came from the NADH, they combine with oxygen to make water. And if the oxygen is not present, then the NADH cannot release the hydrogen because that hydrogen has nowhere to go, and it is not recycled back into NAD+. The electron transport chain complexes pump hydrogen ions from the matrix into the intermembrane space of the mitochondria. So the hydrogen ions therefore become more concentrated in the intermembrane space, creating an electrochemical gradient. You'll remember that nature doesn't like concentration gradients. It wants everything to be equal. So a protein called ATP synthase allows those hydrogen ions to flow down its gradient. It allows them to move from high concentration to low concentration. And that flow of hydrogen ions drives the synthesis of ATP, okay, so the making of it from the ADP, adenosine diphosphate, and phosphate into ATP. This is a process called chemoosmosis. The ATP production is linked to the establishment of a hydrogen ion gradient. So if we don't have that gradient, we can't make the ATP. The ATP then moves out of the mitochondria and is used in the cell for any kind of work that needs to be done. It is then broken down into ADP and organic phosphate. These molecules are returned to the mitochondria for more ATP production. So it's just a big cycle. The ATP gets made, it gets used, broken down into ADP and phosphate, and it is constantly being regenerated in the mitochondria of our cells. The organization and function of the electron transport chain. So here's a diagram of it. Okay, so this is the cristae of the mitochondria. Okay, it's made out of phospholipids and it has proteins embedded in it. And it is their job to pass the electrons from one molecule, from one protein to another using enzymes, and harness that energy. So it's pumping those hydrogens. This is the one that pumps the hydrogens out. Okay, pumps those hydrogens out into the intermembrane space, and the ATP synthase allows the hydrogens to flow back through, and as they flow back through, it makes adenosine triphosphate. And the energy yield from glucose, from glycolysis, we get 2 ATP. The citric acid cycle, we get 2 ATP, and from the electron transport chain, we get between 32 and 34. Okay, so that if it is extremely efficient, every single reaction happens exactly the way that it should, we can get up to 38 total ATP generated from just one glucose. So the energy content, our reactant, the glucose molecule, contains 686 kilocalories. The energy yield of 36 ATP is about 263 kilocalories, so it's only about 39% energy efficient. The rest of that energy is lost in the form of heat. And remember that anytime energy is transferred from one form to another, some energy is generally released in the form of heat. So it's never 100% efficient. So here we have a diagram. We start with glucose. We release 2 ATP by breaking it down into 2 pyruvate molecules. Okay, we break it down into acetyl-CoA. We go through the citric acid cycle, and at each of these steps we are generating NADH and FADH2. Those molecules are then used to make more ATP in the electron transport chain. So we end up with a total of 36 to 38 ATP total per glucose molecule.